Dear children, welcome to biology classes. So, we have completed the first chapter and today we are going to start the new chapter, that is sexual reproduction in flowering plants. So, you know the sex organ of a angiosperm is flower, isn't it? And you have studied the parts of a flower in your smaller classes itself, isn't it? Sepals or calyx, then corolla, that is the units of corolla are petals, then androecium, the units are stamens, and then what the gynoecium, the units are carpels. So like this different parts you have already studied, isn't it? So if you study the parts in detail, each stamen is having a stalk known as filament, okay, and then they will have an anther at the tip. Okay, so if a plant, a stamen is seen, so this is the filament, okay, filament, and this will be the anther, okay, and usually anthers are having two lobes, okay, that is they are bilobed, and each lobe will have two theca, okay, so two theca will be there, that's what it is known as dithecous, okay. So a typical angiosperm anther is bilobed, two lobes are there and in each lobe there will be two theca, okay, so they are known as what, dithecous, okay. Then you can see a groove that is passing through this in between the theca, okay, and in each anther there will be 4 microsporangia, okay. So microsporangia is the part where the spores are produced, isn't it? So the anther is having 4-sided or tetragonal, and they will have 4 microsporangia in each corner, 4 corners, okay, that is 2 in each lobes. These microsporangia, they develop into pollen sacs, okay, in which pollen grains are developing. So when you see the structure of a microsporangia, so microsporangia are found on where? On the corners of what? This bilobed anther. Understood? So each microsporangium, if you see, you can see that they are surrounded by 4 layers, okay, 4 wall layers, that is the epidermis, okay, that is then endothecium, middle layers, and tapetum. Understood? So epidermis, endothecium, and middle layers, their main function is to protect the spores or the sporogenous tissue present inside them, and the tapetum is the nutritive tissue, okay. The tapetum is the innermost one and it nourishes the developing pollen grains. Is it clear? So like this you can see in microsporangium contains 4 wall layers, okay, the epidermis, then comes the endothecium, then comes the middle layers, okay, and inside that means interior to that, what will be there? The tapetum, okay. Tapetum is the nutritive tissue and inside that you can see the sporogenous tissue, okay. So this is the structure of a microsporangium. Okay, so if microsporangium's tissue structure is asked, this much you have to write, okay. So this is a pollen, this is a section of a anther, okay, transverse section of an anther, and if you enlarge and see it like this, okay, this much part you can see this much, okay, that is the different layers, wall layers, 3 wall layers which are responsible for protecting the spores or sporogenous tissue, and tapetum which is giving nourishment for the developing embryo. Understood? So it's very clear now the structure of microsporangium, okay. So how many microsporangiums are there in an anther, in a bilobed anther? 4, okay, and the structure also I said, okay, wall layers, okay, that is the epidermis, endothecium, middle layers, and tapetum, okay, and then inside that what will be there? Sporogenous tissue. Now we will study the process that is microsporogenesis. So what do you mean by microsporogenesis? The formation, okay, the process of formation of microspores from a pollen mother cell through meiosis is called what? Microsporogenesis. Formation of microspore from pollen mother cells, PMC, through what? Meiosis, because these microspores, okay, are haploid, so it should be formed by meiosis. That process is called what? Microsporogenesis. Is it clear? When I told you about the microsporangium, I told wall layers will be there, and in the center there will be some tissues called sporogenous tissues, isn't it? And each of this, listen very carefully, each of the sporogenous tissues, okay, they are capable of what? Giving rise to a microspore tetrad. Understood? So each one of this is potential pollen or microspore mother cell, pollen mother cell. Each of the cells in the sporogenous tissue is capable of producing a pollen grain mother cell or microspore mother cell. Understood? So when these cells are undergoing meiosis and producing 4 cells, they will be arranged in the form of a tetrad. Okay, so microspore mother cells, they undergo meiosis, okay, and produce what? The pollen microspores or pollen grain tetrads. And the anthers, okay, they when they mature, okay, so in the anther, in 4 sides, the sporogenous tissues contains cells, and each cell act as the pollen grain mother cell. They undergo meiosis and they form pollen grain tetrads. Okay, and this pollen grain will get separated, okay, or dissociate from each other and develop into pollen grains. Understood? So like this, in each microsporangium there will be many pollen grains, and when the anther get matured, they dehisce, okay, like this they will dehisce and the pollen grains will be getting dispersed. So you would have heard that pollen grains are the male gametes from your small classes, isn't it? Actually, pollen grains are not the male gametes. What are they? They represent the male gametophyte. Okay, so if you see the structure of a microspore or a pollen grain, okay, it will have a wall having 2 layers, okay, 2 layered walls it is having, and the outer wall layer is known as the exine, okay, exine, and it's made up of a substance known as sporopollenin, don't forget, okay, and it is one of the most resistant organic material. Understood? Sporopollenin. And this is the reason we can get fossils of pollen grains, because this is not easily degraded by the microorganisms and all, and it can withstand high temperature and strong acids and alkali. So this is the reason why pollen grains are found usually as fossils, okay, because microorganisms cannot degrade, temperature will not have any effect, okay, then what, they are resistant to acids and alkali, and enzymes cannot degrade. Understood? So exine is the outer layer with a lot of different ornamentation, okay. You can see here one diagram is here, it's having some spike like, isn't it, ornamentation, and these are pollen grains of some other species. You can see each species exine will be very specific. They will have different articulations. Understood? So this will be very specific to the pollen grain. If you get, we can identify this pollen grain is belonging to which species, because that is very specific to each species. Clear? So exine, a speciality I told, they shows various articulation, different ornamentation, and also it contain a substance known as sporopollenin, and that is very resistant, okay. And then if it is so resistant, then how the pollen tube can germinate, isn't it? When pollination happens, pollen grain has to germinate. So how it is possible? Because there are certain pores, okay, where the sporopollenin is absent. That region is known as germ pores. Okay, so through germ pores only pollen tubes comes out. Understood? During pollen germination. Okay, so the outer layer is known as exine and it contains a substance known as what? Sporopollenin. Now, the inner layer is known as intine. Okay, and the substance that make up this intine are cellulose and pectin. Cellulose, pectin, etc. you can see in there. In the intine. Okay, then cytoplasm will be there, that will be separated or surrounded by a plasma membrane. Then the cell, if you see the pollen grain, if you see, it will have two cells, the vegetative cell and the generative cell. Vegetative cell, if you see, it will be larger in size. Okay, and they will have abundant reserve food materials. Okay, and they will have a irregularly shaped nucleus. It can be asked, how can you differentiate a vegetative cell and a generative cell of a pollen grain? So how you can, vegetative cell will be bigger and will have more food reserves. Okay, and their nucleus is irregularly placed or shaped. Okay, on the other hand, generative cell is small when compared to the vegetative cell and they floats on the cytoplasm of the vegetative cell. Okay, so in this two-celled condition, most that is around 60% of angiosperms, pollen grains are shed. Means how the pollen grains are shed? When the anther dehisces, isn't it? When the anther dehisces, the pollen grains will be dispersed. So in during this dispersion, around 60% of angiosperms, the pollen grains will be dispersed or shed in this two-celled condition. Okay, in the other cells, other species, the generative cell divides mitotically and produce two male gametes. Okay, before the pollen germination. In some species, I told the male gamete production takes place before the pollen shedding or pollen dehiscence, pollen dispersion. Okay, and in some after. Understood? So like this, this is the structure of a pollen grain. So what all things we learned? Do you remember? I told you about the stamens. Stamens parts are what? Anther and filament. Each anther is bilobed and it's having two theca in each lobe. So it is dithecous. Okay, and in each corners you can see what? Microsporangium. What is the structure of microsporangium? It contains wall layers and inside that sporogenous tissue. What are the wall layers? Epidermis, endothelium, middle layers and tapetum. Among this, tapetum is the nutritive tissue and the other layers are they are helping in the protection. So what I told is the structure of stamen consists of what? Or if you see the stamen, they will have a filament and an anther. Anther is bilobe. Two lobes are there and in each lobe there will be two theca. Okay, two theca will be there and in this you can see 4 microsporangium. So in each microsporangium, what you can see? You can see 4 wall layers. What are the different layers? Epidermis, endothelium, middle layers and tapetum. Among these 3 wall layers, they are protective in function and tapetum is responsible for giving nourishment to the developing pollen grains. Okay, so wall layers. Inside the wall layers, there is a group of tissue called sporogenous tissue. This sporogenous tissue is responsible for forming the pollen grains. Pollen grains are formed from sporogenous tissue. Okay, so each of the sporogenous tissue means cells in the sporogenous tissue is capable of forming the pollen grain mother cell, and each pollen grain mother cell undergoes meiosis and produce what? Pollen grains. Understood? Now, the process of formation of microspores from pollen grain mother cell is called what? Microsporogenesis. And in this what happen? Pollen grain mother cell undergo meiosis and produce 4 cells. Okay, 4. Each of this will be capable of forming the pollen grains, and or they are pollen grains, they develop into pollen grains, okay, and after separating from each other, actually when they form after meiosis, they will be in the form of a tetrad, okay, and they separate to form pollen grains. Is it clear? Now these pollen grains, if you see the structure, they will have what the pollen grain is not the gamete, it is represented, or it can be called as what male gametophyte. So what are the components of, or what are the what structure of a pollen grain? It will have a wall layer having two layers, okay, it has a membrane with two layers, that is outer layer is known as exine, and it's made up of a special substance known as sporopollenin, and this is the most resistant organic material that can withstand high temperature, acids, alkali, and no enzymes can degrade them. And there is a space, there will be spaces where there is no sporopollenin, and that place is known as germ pores. What are they? Germ pores. And inside that there will be another layer known as intine, and that is made up of cellulose and pectin, okay. And when you see a pollen grain, when it get matured, they will have usually two cells, okay, that is vegetative cell and generative cell. The difference I already told. So vegetative cell, sometimes what happens in some plants, okay, the generative cell again divide mitotically and form two male gametes. So male gametes are formed in some plants before the pollen shed, okay, before the pollens are dehisced, means dispersed. And in most of the plants, how it will be? After the pollination. You have learned the pollen grain when germinate, then only the generative cell divides to form two cells, okay, two male gametes. So why this pollen grains are important? Because most of the species of pollen grains can produce allergies, okay, allergy disorders like asthma, and it can cause respiratory diseases like asthma, bronchitis, etc. Okay, then the pollen grains they are very rich in nutrients, okay, so it is used for making some food supplements, okay. It has means in recent years we use what pollen grain as tablets or as food supplements, etc. Okay, then likewise many syrups, tablets, etc. are available, okay. Then so pollen grains, some of them they lose the viability after their shed, okay. When they get shed, the pollen grain, some of them they retain their viability. Means what is viability? Their capability to germinate on the stigma, okay. So this will be long for some of the plants, okay. In some plants it will be very long. Pollen grain can retain their viability for long months, okay. And some of them they lose their viability within 30 minutes after their release. So like this, that depends on the species, okay. So this pollen grain also can be stored, okay, just like what semen or sperms, as it is stored under minus 196 degrees centigrade. In liquid nitrogen we can store. So in the same way, pollen grains also can be stored for long time, okay, for many means years, if we store them under this condition. So this kind of condition or this procedure is known as cryopreservation, where we can store the materials, living things, okay, in liquid nitrogen in below 190, means in minus 196 degrees centigrade, low temperature, okay, for many years. So this is known as cryopreservation, and this can be used for storing the pollen grains for many years, okay. So this is the economic importance of what pollen grains, okay. Some of them causes allergy and other respiratory diseases. Some of them I told they can cause what they can cause they are as they are very nutritive. Okay, they are used as food supplements. Okay, and then we talked or we discussed about the pollen viability. Understood? So some of them lose the viability very soon. So this kind of pollen grains can be used for long time by preserving them in low temperature in liquid nitrogen. So this much is very clear to you, I hope. Thank you very much, children.